Myself, Professor Sujata Mahapatra from the Department of Botany, Ramadevi Women's University. I shall be giving a lecture on the topic Fluorescence Microscopy. It is a part of course curriculum both at undergraduate and postgraduate level of this university. Fluorescence Microscope. The first fluorescence microscope was designed by Lehman in the year 1940. And this microscope is a type of light microscope that uses the principle of fluorescence, then reflection. The basic principle is that the specimen is labeled with fluorochrome and the light falling on the fluorochrome is emitted as the fluorescence, and the fluorescence creates the image that can be viewed. Fluorescence and phosphorescence are part of photoluminescence phenomenon. The fluorescent dyes may be organic or inorganic in nature. Fluorescence microscopy has several applications. It is used in cell biology, microbiology, neurobiology to observe the cellular processes, physiological processes, detection of microbial pathogen in patient samples in the field of immunopathology, and the study of nuclear pleomorphism in some forms of cancers in fluorescence in situ hybridization technique too. There are different types of fluorescence microscopy like wide field fluorescence microscopy, confocal fluorescence microscopy, total internal reflection fluorescence microscopy, light sheet fluorescence microscopy, and super resolution fluorescence microscopy. It is a type of modern microscope. The topic of discussion in this lecture is wide field microscopy. It is called as wide field microscopy because wide area of the sample is illuminated and then the image formation occurs. Emission wavelength from different focal planes are collected and there is generation of a two-dimensional image. Epifluorescence microscopy. When the excitation and transmission wavelengths pass through the same objective, it is called as epifluorescence microscopy. It may be of two types. One is reflected epifluorescence microscopy and second is transmitted epifluorescence microscopy. In transmitted epifluorescence microscopy, the specimen has to be very thin because the fluorescence has to pass through the specimen clearly. In case of reflected type, it is a surface phenomenon. The fluorescence is produced after reflection, so thicker specimen may be taken in case of the reflection type of epifluorescence microscopy. Absorption of light at one wavelength and emission at a longer wavelength is called as fluorescence. Some molecules can fluoresce. When they are electronically excited, they release the extra energy in the form of fluorescence or phosphorescence, and these molecules are called as fluorochromes. And fluorochromes are used to stain the specimen in fluorescence microscopy. This is the mechanism of fluorescence. This is the atomic nucleus and the orbit of the electron. This is the ground state electron. When it absorbs light of certain wavelength, it is excited to the next level or the excited state, it becomes the excited state electron. When it gets back to the ground state, it releases extra energy in the form of fluorescence or phosphorescence depending upon the level of excitation. So this is fluorescence and the fluorescence emitted by this electron is of a longer wavelength and less energy than the incident wavelength. Incident wavelength is shorter and the fluorescence wavelength is longer. Incident energy is higher and fluorescence energy is lower. When the excited electron returns to the ground state, it produces either fluorescence or phosphorescence. The difference is fluorescence is produced by the electron when it is in the singlet excited state, and phosphorescence is produced when the electron from the triplet excited state returns to the ground state. These are the examples of different fluorescence dyes, and each dye is specifically used to stain a particular biomolecule or an organelle. DAPI, acridine orange, and fluorescein can be used. GFP is an example of autofluorescence biological molecule. So this is organic in nature, and each fluorescent dye has its own excitation maxima and emission maxima, and accordingly, depending upon the sample, the choice of the fluorescent dye is made. The concept of fluorescence was given in the year 1852 by George Gabriel Stokes, and according to his view, there is a difference between the positions of the band maxima of the absorption and the emission spectra of the same electronic transition. It is called a Stokes shift after the name of the scientist. So there is a shift of the incident light to lower energy when it is absorbed by a fluorescent molecule. That is why the emitted light is of a longer wavelength than the incident light. So this shifting of the emission is called as Stokes shift. This graph shows the Stokes shift. This is the absorption maximum and this is the emission maximum. So there is a shift of the maximum from a lower wavelength to higher wavelength, or from higher energy to lower energy state. The basic components of the light microscope involve the light source, excitation filter, dichroic mirror, objective, sample stage, emission filter, and ocular or the detector. The light source is very important, and the light source must be able to produce high intensity light, and the light must be near monochromatic. The intensity should be such that it will excite a fluorophore. Mercury arc lamp can be used, high powered LED can be used, laser light, supercontinuum sources may be used, but the mercury arc lamp is a very good choice because it emits high luminescence and it produces high luminance radiation both in the UV and visible wavelengths. Excitation filters are made up of optical fiberglass. They allow only the wavelengths which excite the fluorophore and held back all the unwanted light. It selects short wavelength light which is of high energy. There are different types of excitation filters like band pass filter, short pass filter, and long pass filter. The band pass filter, it allows the selected frequencies and holds back all the unwanted frequencies. Short pass filter permits the frequency shorter than the cutoff frequency, and long pass filter permits frequencies longer than the cutoff frequency. In the sample, some areas are stained with the fluorochrome. The function of the emission filter is to catch the fluorescence that is emitted by the fluorochromes from the sample. It selects the in-focus wavelength and heads back the out-of-focus light. So it cuts off or absorbs the unwanted excitation wavelength from the fluorophore tagged sample, and it permits only a narrow band of wavelength which corresponds to the emission wavelength of the fluorophore. And this produces the final image in the microscope. Dichromatic mirror or beam splitter, it reflects the light that comes through the excitation filter, and this reflected light is directed towards the specimen after passing through the objective. So the reflected light first passes through the objective, then it falls on the specimen. The emission light that comes out from the specimen is also collected by the objective and reflected by the dichromatic mirror. After that, it passes to the emission filter. So, dichromatic mirror has important function in directing the excited light and the emission light. The objective of the fluorescence microscope is made up of special quality glass or quartz, and it transmits light both in the UV as well as infrared wavelengths. This is the interior of the optical block or the filter cube that holds all the different types of filters. Inside this optical block, there is dichromatic mirror, there is barrier filter, and there is provision for the entry of the incoming light waves. So it is a single unit that houses all the types of filters. Detector for fluorescence microscopy may be simple ocular lens or a digitalized camera like the charged copper camera or cooled CCD camera. Cooled CCD camera are better because they reduce the noise of the image. This is the image of cell division showing the cell plate formation under the ordinary light microscope and under the fluorescence microscope. So fluorescence microscope is very versatile in the imaging of different types of samples. This is the picture of each cell under the fluorescence microscope. The cell membrane can be clearly seen. And this is the under the ordinary microscope. So the glowing cells are clearly seen under the fluorescence microscope. This is the operating principle and path of light of a fluorescence microscope. Short wavelength light of high intensity is selected by the excitation filter, and after that it falls on the dichromatic beam splitter. The dichromatic beam splitter selects certain wavelengths which will excite the fluorophore and directs those wavelengths to the objective. The wavelength passing through the objective, they fall on the specimen and excite the fluorophores. Fluorescence emitted by the fluorophores and both in focus as well as out of the focus light passes to the objective, then again to the dichromatic mirror. From the dichromatic mirror, the light is reflected. This is the reflected fluorescence wavelength. It is selected by the emission filter and Only the in-focus light passes through the detector pinhole, and then image is created by the ocular. This image shows the principle of excitation and emission in a fluorescence microscope. The excitation filter, it selects the particular wavelength, and excitation UV light is selected. In this example, the UV light is selected and it is focused on the specimen, which is labeled by fluorochromes. The fluorochromes get excited and emit fluorescence. The fluorescence light, both in focus and out of focus light, pass through the barrier filter. The barrier filter, it holds back the out of focus light and allows the in focus light that is cut by the ocular. And then it passes to the camera or can be viewed with eye. These are the components of a wide field microscope. Here the sample is here. The mercury vapor lamp produces polychromatic light, and the light is selected and falls on the sample. The fluorescence wavelengths emitted by the sample are directed by the dichromatic mirror, and they are directed to the CCD camera that captures and creates the image. This is the diagram of a epi-illumination reflecting microscope. In the reflecting microscope, principle of reflection is used. So the sample surface fluoresces the light, and the fluorescent emitted from the surface of the sample, it creates the image. So the sample is held on the stage, and the light source creates the light that passes through the excitation filter. Excited light is directed by the dichromatic beam splitter, passes to the objective, then through the objective it comes to the specimen and falls on the specimen. The fluorescence chemicals are excited, they emit the excitation wavelength. The excitation wavelength is again passes through the dichromatic mirror and then passes through the barrier filter or the emission filter, and then passes to the CCD camera or through the ocular, it can be observed. This is a schematic diagram of the transmitted fluorescence microscope. Here the specimen is very thinly prepared and it is labeled with the fluorochromes. The light comes from below the specimen, passes through the fluorochrome labeled specimen, passes to the objective and creates the image. So as the principle of transmittance is used, this is called as epi-illumination transmitted microscope. This is the experimental setup that shows a smartphone converted to a fluorescence microscope. If it is successful, it can be very useful for biomedical analysis. The modern day fluorescence microscopes, specifically wide field microscopy, has very sophisticated electronic imaging devices attached to it. The optical components are sophisticated. There is computer control of the focusing, the adjustment of filters, etc. And it is a very powerful tool for the field of medical science, microbiology, and even for the detection of the cancer. Thank you.